Hello everyone and welcome to Thermo Fisher Scientific webinar series on Sanger's sequencing and fragment analysis made easy, corresponding with the launch of Applied Biosystems' new SeqStudio genetic analyzer. Today we present the third topic in the series, enabling neurological disease research via DNA fragment analysis on the SeqStudio genetic analyzer, presented by Dr. Gary Latham, Senior Vice President, Research Development at Asuragen. I'm Susie Valdez, and I'll be your moderator for this educational webinar presented by LabRoots and sponsored by Thermo Fisher Scientific. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the green Q&A button located at the lower left of the presentation window. Type your questions into the box that appear on the screen, and we'll answer as many questions as we have time for at the end of the presentation. Also, notice you're viewing this presentation in a slide window. To enlarge that window, just click on the screen icon at the lower right. If you have any trouble seeing or hearing this presentation, click on the support button or use the Q&A button to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button at the bottom left-hand corner and follow the process for obtaining your credits. I'd now like to introduce today's speaker, Dr. Gary Latham. Dr. Latham is a Senior Vice President of R&D for Asuragen, a molecular diagnostics company focused on genetic and oncology. Gary joined Asuragen when it founded in 2006 and currently oversees the company's research, assay development, partnered R&D, and bioinformatics team. He guided the development of AmplideX PCR technology for Fragile X syndrome and other DNA repeat disorders, and DNA and RNA-based QuantideX NGS products for oncology applications. Gary is an inventor on 12 issued U.S. patents, has received over $13 million in research grants for the NIH and Cancer Prevention and Research Institute of Texas, and has authored over 40 peer-reviewed publications. Please join me in welcoming Dr. Gary Latham. I will now presentation over to him.
All right, Susie, thank you for the introduction. It's a real pleasure to be here, and I'd like to thank our partner, Thermo Fisher, for the opportunity to present within this webinar series. I'd like to talk about some novel technology that we've developed at Asuragen and how it may be combined with the new SeqStudio genetic analyzer. And I'd like to begin by talking about three problems that relate to challenging genetic analyses and propose a potentially unifying solution. So problem number one, how do you reliably PCR amplify some of the toughest stretches of the human genome to advance clinical research? Problem number two, how do you simply and easily resolve these PCR products to generate high definition insights? And problem number three, how can you operationalize a straightforward workflow that can interrogate these challenging loci to enable routine analysis? And the solution, I believe, is to pair a processive PCR with a high-performance platform, the right innovation with the right instrument. But before I get there, I'd like to first talk about some of these really hard to amplify patches of the human genome, this dark DNA, and how it is emerging and has emerged as important regulatory elements in gene expression, and in many cases as bona fide biomarkers of inherited disease, with implications that range from neurological disorders to reproductive health. So with my disclosures in the background, let me begin by saying that the known genome is not the whole genome, nor is it the whole story for markers of genetic disease. And although we're nearly 15 years past the official close of the Human Genome Project, we still don't have a fully complete end-to-end sequence of our genome. And although estimates vary, we currently have about 5% of our genome sequence has defied conventional sequencing technology and has remained dark. And although we don't know the exact sequence of this dark DNA, we do know that it's comprised of mostly repetitive DNA. But we have an opportunity now with new technology to be able to shine a light on this dark DNA and to understand the specific nature of this sequence. Because we have long read NGS that's now available that can read through these repeat elements and the flanking segments to be able to contextualize repeats. And we have fit-for-purpose bioinformatic analyses that can address some of the special challenges of repeat DNA to be able to define the sequence. And it's not a surprise that this remaining recalcitrant dark DNA is repetitive in nature, because in fact, half of our genome is comprised of repetitive DNA. And we can bucket that into about 3 different categories. There's a large chunk that's retrotransposons, another component that's DNA transposons, and a third category that's STRs, or short tandem repeats. And these are motifs of 1 to 6 or so nucleotides that are scattered throughout the genome and are very abundant. For example, we know that there are at least 1 million STR elements in the genome. And just looking at the real estate that they occupy, they consume about 10 times the land mass of the genome compared to SNPs, and even twice that of our coding genome. In addition to being highly abundant, they're also highly plastic. The per mutation or per generation mutation rate associated with STRs is, or can be, orders of magnitude greater than SNPs. And that means that they can be highly variable in different populations. Further, because they can occupy multiple length states, they are multi-allelic. So unlike SNPs, which are typically binary, it's a C or it's a T, with a repeating element, it can be different lengths and thus can function over a digital continuum of possible states that may have an ability to fine-tune any functional impact they have. And when we speak of function, we should also think about their potential roles in gene expression, because 20% of genes have at least one pure STR within about 1 to 2 kb of transcriptional start site. So these repeating elements are poised in the regulatory regions of genes that suggest they can have an important functional role in the cell and may have links to disease. So that's the general lay of the land, the demographics of STRs, why they may be important to pay attention to. But in addition, we also have specificity from single gene studies that suggest how they may modulate function, and particularly gene expression. So for example, bidirectional transcription, we know, can be influenced by repeats, where increases in the number of repeats can alter the level of sense or antisense transcription. And a good example of this is SCA8, spinocerebellar ataxia type 8. This is a disorder with a pathogenic repeat expansion, where interestingly, the sense transcript can be translated into a toxic poly-Q protein, whereas the antisense transcript, which contains the repeat, can be a sink to bind the splicing factor muscleblind and trigger larger scale splicing defects within the cell. Now, in addition, we have also quite a bit of evidence of more direct effects on RNA splicing as a way repeats can affect expression. For example, it's been known for years now that the TG repeat in intron 8 of the CFTR gene alters splicing at exon 9, which then reveals itself in some cases in a clinical phenotype of male infertility with congenital bilateral absence of the vas deferens and non-classic CF. Further, epigenetic modifications are well known with pathogenic repeat expansions. Fragile X, reduced ataxia are two such examples where when the repeat exceeds a certain threshold, then you see repressive chromatin marks laid down and silencing of the gene, which is directly associated with the consequence of the disorder. And in addition, we also know that as repeats get tangled within the transcription bubble, they can also bind to the DNA within that bubble and create R loops that themselves can have an impact on epigenetic modification. And so in general, what we've seen in more and more cases is that increases or decreases in the number of repeats can affect gene expression by increasing or decreasing transcription. And although that's very clear from these individual gene studies, now we're beginning to look with the right tools, both informatically and from the standpoint of methodology and technology, genome-wide, so that we can canvas the genome, we can look at these STRs and understand their functional impact. And a terrific paper came out last year from Melissa Jemreg and Nina Ehrlich in Nature Genetics that showed that they could identify more than 2,000 ESTRs whose links were linearly associated with nearby gene expression and could explain 10 to 15% of cis heritability attributed to common SNPs. And so repeats may in fact be a way to help understand the, quote, missing heritability that's been associated with some diseases. So in addition, it's useful to think about the functional impact of STRs in terms of how intolerant or not they may be to mutation. Now, on one hand, I mentioned that STRs can be much more plastic than SNPs by comparison, but that doesn't mean that they're tolerant of mutations at every locus, which then suggests function within their role in the genome. And this study, which just came out within the last month, looked specifically at whether STRs were genetically constrained. And the way this study was conducted was to take data from the GTEx database and look across different tissues and compare the impact of STRs with the top 20% expressed genes compared to the bottom 80%. And ask the question, are the top expressed genes more genetically constrained with respect to STRs? Do they differ from one tissue to the next? And what they found was that they are constrained. In almost every tissue, the level of genetic constraint was highest in the most highly expressed genes. But what was really fascinating about this study was the genes that were most constrained were abundant within the brain compared to any other tissue. And that's tantalizing to think about, given that many of the canonical repeat expansion disorders are pathogenic in the brain and brain-related tissues. So let's recap. We have repeats that are dark in sequence, but are increasingly discoverable thanks to long-read next-generation sequencing as a discovery tool. We have repeats that are dark in function, but are increasingly knowable because our understanding of the linkage between STRs, gene expression, and also potential association with disease. And the coup de grace is that we have more than 30 known disorders that are associated with repeat expansions. And these disorders represent an overabundance of disorders of the brain, neurodevelopmental, neurodegenerative, and neuromuscular. And they can represent expansions in introns or in exons, like the poly-Q disorders, or in 3' or 5' UTRs. I'd like to put a spotlight on a handful of these today, 5 of these, and talk specifically about how we've developed PCR technology to be able to consistently read through these repeats, whether they're AT-rich or GC-rich, and how those products can be interpreted on the right platform, like a genetic analyzer, capillary electrophoresis, to generate definitive results for clinical research. And I'll talk about 4 of these pathogenic disorders, 1 emerging risk marker in Alzheimer's disease, And these represent 3 products that we've developed that are on market from Asuragen and 2 that are in development. So let's start with Fragile X. So Fragile X is caused by a CGG repeat expansion in the 5' UTR, and more modest repeat expansions are known as premutations, and those have 55 to 200 repeats. And they're relatively common in the population. About 1 in 200 women and about 1 in 400 men are premutation carriers. And because of this incidence, Fragile X sits aside SMA and cystic fibrosis and a handful of others as a common target for carrier screening. But in addition, carriers are at risk for a couple of different Fragile X-associated disorders. One of these is FAXTAS, or Fragile X-associated tremor and ataxia syndrome. This is an age-onset neurodegenerative disorder whose symptomology is often confused with Parkinson's disease. And then in women, FAXPOI, which is Fragile X-associated premature ovarian or primary ovarian insufficiency. And FAXPOI affects about 20 to 25% of premutation women, and many others that are carriers may have milder forms of ovarian dysfunction. So this is a very significant disorder within premutation carriers. More extreme amplification or expansion of the CGG region in FMR1 beyond 200 repeats is known as a full mutation, and that's the cause of Fragile X syndrome, which is the most common known genetic cause of autism and a major neurodevelopmental disorder. It occurs in about 1 in 5,000 boys and girls. And it's important then, across this spectrum, to be able to have technology that can accurately flag full mutation expansions, but then also accurately assign the size of the CGG repeat within the premutation or other ranges. And this is important for several reasons. One, you need to be able to differentiate true homozygosity in females from apparent homozygosity. In other words, it's advantageous to be able to have even single CGG repeat resolution so that you can tell the difference between a 29 and a 30 versus a homozygous 29-29, or a 29 that's heterozygous with a full mutation that using conventional PCR technology might drop out. In addition, because categorical boundaries are defined by a single repeat, it's important to be able to have accuracy for the CGG numbers so you don't miscategorize the sample. And then finally, in the case of premutation women who are at risk in transmitting a premutation allele that can expand to a full mutation in the next generation, it's important to accurately size the CGG repeat, because even a plus or minus 2 CGG difference in premutation women can translate to a 20% differential risk for expansion in the next generation. So you want to make sure that you get that right. So the question is, how do we do that? How do we deal with the fact that this is a CGG repeat expansion? By definition, it's 100% GC. How do we deal with the fact that we need to identify more than 200 repeats of these? And some of these repeats can be even greater than 1,000 or more in number, but then also have the finesse to be able to look at precisely the number of repeats that may be present in a sample for the reasons that I described. And the way we do that is using a novel PCR reagent set based on optimized chemistry, primer design, and thermocycling to be able to generate products that can navigate this high GC rich content. So just to give you an overview, the way we do this is we have a 3-primer design. We have 2 outward gene-specific primers, one of which is labeled with a fluorophore so we can resolve it on a platform downstream. And then we use a 3rd primer, which is a repeat primed element. So this is a primer that's complementary to the CGG repeat. It lays down and binds to each and every one of these repeat elements and combines with a reverse primer to create a multiplicity of products that can be resolved. So what this looks like on a capillary electrophoresis for fragment sizing is that you get 2 categories of peaks, the series of repeat prime peaks on the left, and then a gene-specific peak on the right. So let's break this down. So what it looks like specifically is the combination of the 2 outward gene-specific primers will generate a full-length peak that spans the repeat. And even if constrained by the sizing of the platform, is not constrained by the PCR. So we can make very, very long products here. And then the contrast to that is a complementary signature. It's generated from the combination of each of these repeat primed primers with the reverse primer to create a series of different peaks that are separated by 3 nucleotides or 1 repeat unit. And this can telegraph a full mutation no matter how long it may be, because we've optimized the primer concentrations to be able to generate this repeat prime pattern all the way out to 200 repeats or all the way out to a full mutation. And it gives you the resolution to be able to look at mosaicism that may exist within the target in combination with gene-specific peaks. So there's tremendous complementarity here to make sure that you never miss a full mutation, but then you also have sizing accuracy through the gene-specific peaks and the supporting repeat prime peaks relative to a calibrating ladder to be able to convert mobility into CGG repeat number. So all the data I'm going to show you from here on out is on the SeqStudio, with one exception, and I'll point that out when I get there. So let's take a look at what the data look like off the platform. This is a challenging sample that I'm showing an electrophoregram for. It's a pool of more than half a dozen different alleles ranging from normal with a low of 18 CGGs all the way out to a full mutation of well over 200. In fact, in this sample, it's over 1,000 CGG repeats. And so this sample tells us several things. First of all, it tells us that we can accurately size because the numbers that are labeled for these peaks are in fact the CGG numbers that we know from orthogonal methodologies that represent the corresponding CGG numbers. Secondly, it covers a categorical range from normal to full mutation. And third, we can even see very closely sized repeats, like 30 or 32. So a separation of 2 CGGs we can pick up. I'll show you an example in just a minute of one. So the platform's doing what it should be doing. We're able to accommodate the breadth of both sizing for longer repeats as well as the precision and accuracy for shorter repeats. Now these are data that were generated via GeneMapper 5, but we've also shown proof of concept for how these data can be ported onto our AmplideX PCR-CE FMR1 reporter software. And this is a fit-for-purpose software that provides project management and data management for FMR1 molecular characterization. It provides automated sample QC and, importantly, push-button genotyping so that you can nearly instantaneously analyze hundreds of sample genotypes. And so our approach, particularly here in the US, has been with RUO products that incorporate wetware, hardware, and software. Wetware, the right reagents, in this case for this challenging locus, for PCR, that can be combined with the right hardware, and we've already established this assay with Thermo Fisher's 3730, 3500, and 3130 instruments, and software. In this case, assay-specific software that gives you the confidence of reliable readouts from primary electropherograms and creates a system solution that's end-to-end. And so we're excited to follow this then up with some of the preliminary data that we have from the SeqStudio. So whether it's Fragile X or some of these other pathogenic repeat expansion disorders like Friedreich's ataxia, myotonic dystrophy type 1 and 2, C9orf72, which is affiliated with ALS and FTD, all of these disorders have in common sequence variations from the primary repeat expansion that can occur in the repeat tract itself or in neighboring sequence. And so in Fragile X, that sequence variation is AGG interruptions. And so these live within the repeat tract, and they act as stability anchors for expansion of the CGG repeat in the next generation. So they're important to identify because they add another dimension to the risk equation associated with expansion from mother to child. So I gave you the example earlier that if you have a repeat that's in the mid-range of a premutation, plus or minus 2 CGGs can create a 20% differential risk. Well, if you take that same mid-range premutation allele and compare 2 AGGs versus 0 AGGs, the 2 AGG sample will be less than 10% risk, and the 0 AGG sample will be more than 70% risk. So you can see as much as a 10-fold difference as a function of AGG for a given CGG length. So this assay doesn't definitively reconcile the AGG in sequence context and AGG number by itself. For that purpose, we have a separate assay that we run in Asuragen's clinical laboratory known as Expansion Interpreter, but it does flag these AGG interruptions, and it provides another data signature that can be very useful in the characterization of FMR1 alleles. So how does this work? So it's still a repeat prime PCR approach, the same assay I described to you previously. The difference is that when those repeat primers encounter an AGG element, they don't hybridize because the assay has been calibrated to prevent that. So this creates a footprint or a dip in the signal intensity in the pattern that's a telltale sign of an AGG interruption. So what it looks like, this is a sample of a 2930, and again, this is SeqStudio data. You can see the 2 peaks here. One of these is a 29 and a 30. This is very different than what you'd see for a 29 or for a 30 homozygous. SeqStudio differentiates those very nicely. Then we go into the repeat prime pattern, and as those primers kick in, you start to generate a series of these repeat prime peaks. However, once the primer runs into the AGG element, it doesn't generate a product because it won't hybridize. So now, within that mobility space, you see a gap, and that gap is associated with the AGG. Then the assay picks back up, and when you run out through that AGG, you pick up the corresponding CGG elements again and can carry forward with the interpretation. There's actually another AGG here that I didn't mark for simplicity purposes, but you get the idea. The assay can give you really subtle differences in signal intensity that can be very informative in a number of different ways. And for those to be fully tapped into, you have to have a high-resolution platform like this type of instrument. So what I mentioned previously was that the ability to flag these large expansions can accommodate a great deal of the need for how you resolve molecular characterization of FMR1. And in fact, using this primary AmplideX PCR FMR1 reagent set, you can reduce the level of reflexing to, say, a southern blot for long expansions by 10 to 50-fold, but you can't completely eliminate it. And that's because one other important molecular characteristic of expanded alleles needs to be adjudicated, and that is the methylation state. So in this particular case, what we've done is we've leveraged the long-read PCR for the AmplideX technology to be able to read out allele-specific methylation. And we do that using a restriction in a nuclease that's sensitive to methylation marks. So how this works is that we have, say, a full mutational allele, and it may be methylated, as many of them are, and we split that original sample into 2 aliquots. We carry out a controlled digestion, and we carry out a digestion with HEPA2, which again will be sensitive to the methylation state. If the sample is methylated, then HEPA2 sites are protected, and as a result, you'll be able to make a full-length amplicon. However, if either of those 2 HEPA2 sites are unmethylated, then the restriction in the nuclease will cut, and it will break the template in a way that will prevent a product from being formed. And so by dividing the sample into 2 reactions and doing a control plus a HEPA2 digestion, and then tagging those 2 different reactions with 2 different dyes on that reverse primer, we can create a composite signature that gives us the sizing of each of the alleles and a comparison of the fraction of methylation for those alleles. And so I'm showing here is actually the same profile for the composite of different FMR1 alleles that was shown a few slides ago, from 18 all the way out to greater than 200 full mutation, with the addition of a couple of controls that are incorporated within this reaction. But you start to see the benefit of being able to assess methylation when you split out the 2 color channels and then being able to do relative quantification of each of these peaks to assess the fraction that's methylated. So this is tapping into now 3 different color channels within the SeqStudio, because we have a ladder, a sizing ladder in one channel in rocks, and then we have these 2 other color channels that we use to resolve allele-specific methylation. The peaks that are circled here in the control condition are those that are completely obviated in the methylation-sensitive digestion. So these are unmethylated alleles, and thus they're digested away, so you don't get a corresponding peak in the green trace. Those that are retained, either in full or in part, are methylated to some degree, and the relative peak intensities of these individual amplicons then allow us to quantify what that methylation fraction is. I will point out this is an unusual sample because the full mutation is unmethylated in this case. We see those rarely, but we do see those. It's a function of the nature of this particular sample. But normally what we would expect to see with a full mutation is that it's fully methylated. But the important point is that we can then quantify that relative methylation as a function of each of the alleles. Now, to push the envelope a little bit further, one of the other things we did was to look at the potential to be able to resolve multiple repeat expansion targets in a single tube assay and a single injection. So in this case, we've looked at FMR1 and FMR2. FMR2 is a putative transcriptional activator. It's a gene that sits very close to FMR1 in the genome. This is a premutation FMR2 allele, where large expansions of FMR2, like FMR1, are associated with intellectual disability. We've tagged the primers that are specific to these 2 different gene loci with different dyes so that we can resolve them, just like I described with methylation PCR. And in this case, you can see the premutation expansion that's associated with FMR2. This sample that's in the tube here from the single tube multiplex is normal for FMR1, so you get the characteristic pattern. You can also see the AGG dips in that green trace. And so this shows that we can, in fact, multiplex different repeat expansion targets, even when they do represent really difficult to amplify GC character when we use the right instrument. So that's a high degree of challenge working through FMR1. But there's even a stiffer challenge, and that's with a different pathogenic expansion, which is a G4C2 expansion in intron of C9orf72. So C9orf72 was identified by a couple of groups in 2011. It's a genetic bridge that links ALS and frontotemporal dementia. Repeat expansions at this locus are associated with about 5 to 10% of sporadic ALS and FTD, and about 35 to 40% of familial disease. But the expansions can be in the thousands. So this is really hard to amplify. So how do we do that? Well, we've taken the same approach that I described before with the FMR1 target and fitted it and optimized it for this particular repeat expansion. And what the profile looks like, very similar to what I showed you with Fragile X, is that this is an expanded sample where the normal allele is 2 repeats and the expanded allele goes out beyond 145. If we do a zoom in on that, you can see the peak pileup that's associated with that greater than 145. And it's important to note that, much like I had mentioned with Fragile X, that the ability to amplify longer products is within the PCR. The limitation at 145 repeats in this case is only from the platform. It's not from the PCR. And just to give you a further indication of that, you can see the agarose gel image here, which shows up to 1,000 hexanucleotide repeats that we can detect. Now, also like Fragile X with the AGG interruption, there are sequence variations that are associated with C9orf72, and I want to give you one example of that. Now, this isn't an interruption within the G4C2 hexanucleotide repeat. It's actually a little bit outside of that region. So these 2 traces that you see, the top one is a 1344, and you can see the corresponding first repeat peak that's marked there with a blue arrow. The sample below it, that electrophoregram represents 17/expanded sample. But note that the first repeat peak is now shifted over to the left. It's marked in red. And if we line up those 2 traces, you can see it's shifted by about 4 peaks, a little bit less than 4 peaks. And since the peaks themselves are distanced by 6 nucleotides, slightly less than 4 peaks should represent slightly less than 24 base pairs. And since they're shorter, you would expect that this may represent a deletion. And in fact, that's exactly what we observed. When we sequence this region by Sanger sequencing, we found that it's a 23 base pair deletion. So the assay has the capability to indicate these types of variations, and with the right platform, you can get a very good sense of any offset and changes associated with insertions and deletions, which can be very powerful for getting a lot of information from one simple workflow. So one other feature of the SeqStudio that I wanted to highlight is the fact that you can optimize the assay for sensitivity or resolution based on your specific requirements using run voltage. So for example, if we take an expanded C9ORF72 sample, as shown at the top here, and we run it at 4,000 volts, we can count out to about 175 repeats, a little more than 145 because the baseline conditions are a little bit different than the ones I just showed you. But if we start cranking up the run voltage to 6,000, 7,000, 8,000, all the way up to 9,000, what you see is we begin to pull in the number of repeats that we can count from 175 all the way out to 120. However, we are increasing the sensitivity of the assay because we're also drawing in the pilot peak, and we're aggregating these longer products into one peak that can give you a much stronger signal. So it depends on what you're trying to accomplish with your assay, to be able to favor individual high-resolution sizing out to some very long distance, or whether you want sensitivity beyond a certain threshold. The assay has the capability to be able to do that. And because you can run many different samples on one plate with different run modules, which is exactly what we did here, you have a tremendous amount of flexibility to be able to quickly isolate optimal conditions to suit the assay at hand. So that's GC-rich. What about the other side? What about AT-rich sequence? So TOM40 is a gene that encodes a mitochondrial membrane translocase that has an intronic poly-T homopolymer repeat. And this repeat has been linked with the risk of age of onset in Alzheimer's disease, as well as the rate of cognitive decline in elderly individuals. In research papers, the poly-T link has been categorized in one of 3 different linked groups, either short, long, or very long. And here again, it's important to be able to size accurately. So not only do we have to negotiate very AT-rich sequence here, very different story from GC-rich, we also have to be able to tell the difference between 29 repeats and 30 repeats, because they represent different characters. So a single repeat here is a single nucleotide. Now, if you use routine PCR conditions to amplify this particular region, you get a profile that looks like this. But I have no idea whether this is one allele or it's 2 alleles or it has mosaicism or what may be happening here. But again, if we apply AmplideX chemistry to this to be able to provide very resolute and defined products, now we can tell exactly what this is. The 2 highest peaks are in fact the 2 genotypes that are associated with the 2 corresponding alleles, 34T, 36T. Separation of 2 T's can be made very easily here. This data was generated on the 3500 CE, but we've also run it on the SeqStudio, and we get essentially the same result. We have this very characteristic peak morphology for these samples that give you single base pair resolution, which is exactly what we needed at this locus. And this is a basis for a research product that we launched fairly recently and can help standardize this type of assessment from one lab to the next to harmonize results and accelerate progress. So I'll wrap up here with 2 prototype assays. One of these is at the DMDK locus, which is a gene that has a 3' CPG expansion. And that expansion is linked to myotonic dystrophy type 1. So the clinical threshold for mild disease is about 50 repeats. The sample shown here is a consensus sample that was reported in a publication a couple years ago from a Ream study of a number of different laboratories that was flagged as a 1275 CPG. We pick up the 1275. We also pick up a mosaic at 63. That's repeatable. We believe that's real. So we believe the assay is performing as expected, and it's very sensitive. What about a larger expansion? This one goes out to beyond 200, which would suggest a more severe disease. By comparison, this is a residual clinical sample that was provided to us from Greenwood Genetics Center. And you can see the trace on the SeqStudio. Again, the ability to see the longer products is within the PCR, just outside of the reach of the platform in this case. And what that looks like is shown here in the agarose gel out to 1100 CTGs, which we can very clearly differentiate here. So we have the capability to reach out for quite a bit of distance to be able to size these alleles, depending on how we resolve them downstream. And then the last case is with Huntington's disease. This is at HTT, it's in the exon, it's a CAG, clinical threshold for disease is around 40 repeats, and I'm showing an example here of a 1570. We have no trouble picking up the 70. In fact, we've run up to 250 with this particular repeat expansion and been able to detect it just fine. So both of these show that the SeqStudio has a potential to be able to accommodate these number of repeats. So I'm going to wrap up here by saying that we have moved on from the GWAS era and arrays to look at SNPs and understand associations to go into other categories of sequence that can be meaningful at the level of cell function and disease. And that includes repetitive elements. Historically, repetitive DNA has been thought to be phenotypically neutral. But now we know that is simply not the case. We know that these, in many cases, are elements that are increasingly discoverable, knowable, and have diagnostic implications. And using the right tool for the right job, long-read NGS, a fantastic discovery tool. However, if you want to operationalize a workflow that can look at known repeats and be able to provide definitive information for their molecular characterization, the combination of PCR using AmplideX chemistries with capillary electrophoresis, the 3730, the 3500, the 3130, and now we see a lot of promise around the SeqStudio is a way to address that particular need. And so that answers the three problems that I posed at the beginning. We do have reliable PCR chemistries that can amplify these really challenging stretches of the genome. We do have small footprint, readily accessible and user-friendly instrumentation that can resolve these PCR products. And we do have workflows that can put these together to generate real insight on the back end. So I'll stop there and open the floor for questions.
Thank you so much, Dr. Latham, for that informative presentation. We will now start the live Q&A portion of this webinar. If you have any questions you'd like to ask, please do so now. Just click on that green Q&A button at the lower left of your presentation window, type the question into the box that appear on the screen, and click that send button. We'll answer as many questions as we have time for. So let's take a look at our questions from our audience member. We have wonderful audience members today. Our first question, Dr. Latham, is what was the size range of fragments that you analyzed on the SeqStudio?
Okay, sorry for the delay there. So the question was, what was the size range of fragments that were analyzed on the SeqStudio? So we evaluated the instrument with amplicons that could be accurately sized from just over 100 base pairs to over 1,000 base pairs. And I'll say that these results were both appropriate for and consistent with the target specifications for the various repeat primed assays that I discussed, without the need for further analysis of the products based on those specifications. I mean, to be clear, longer products, I mean, even those that are up to several kb, like the 1,000-ish G4C2 repeat in C9 or 72 that I showed, are detected on the SeqStudio, but they pack into the pilot peak. And this actually has a benefit. The benefit is an improving sensitivity, which is ideal for, say, full mutations of FMR1, where when you go beyond 200 repeats, there's no indication of genotype-phenotype correlations that are useful. So this pileup that can happen can be very beneficial for the assay because it can increase that sensitivity. Now, if there is interest in sizing some of these really long products, then, as I mentioned, you can potentially put them onto agarose gel electrophoresis. But for the most critical step, for all of the assays that I discussed, it would be to achieve high resolution in the 100 to 1,000 base pair range, and that's exactly what the SeqStudio delivered in our evaluation.
Thank you so much, Dr. Latham. Our next question from our audience is, what was the detection sensitivity of low abundance repeat containing amplicons, and what are the implications for the detection of low level mosaicism in genetic samples?
So in the course of our evaluation, we easily detected a 5% mass fraction, say with FMR1. In fact, we achieved as low as a 1% equivalent of full mutation as an admixture using a 940 CGG FMR1 cell line in a background of an expanded cell line sample. And we would expect that the sensitivity for lesser repeats in the premutation range would be similar. We've done comparable studies against some of the other targets with the other genetic analyzers in the ABI-CE family, and have seen results that are very consistent with those types of findings. So we expect that being able to look at a few percent of cell equivalents based on the sensitivity of the instrument is achievable.
Thank you so much, Dr. Latham. And thank you again for our live audience participation. We have some wonderful questions coming in. Our next question is, you spoke of making a process around these repeat detection assays. Can you give a sense of how long a single analysis takes, or how many discrete samples can be analyzed in a day from raw samples to completed results?
So that's a good question, and it really speaks directly to the nature of the specific assay and what the target locus and specifications might need to be for the given purpose. So some of the targets that I mentioned are relevant for large-scale sample analyses, and in those cases you really would want to be able to look at many samples at a time and process those in a relatively high throughput manner. In terms of how long it takes to be able to do an analysis like that, and the assays themselves are highly scalable, so you have the capability to run these at larger densities. And in terms of workflow, most of these assays take less than about an hour of hands-on time to run through entirely, because you do your PCR, you generate your products, depending on the target. And some of these highly repetitive assays have relatively short targets. They might be, again, as I mentioned, only 100 or a little bit more in terms of base pairs, in which case you can do relatively short run times to be able to resolve them. And you would need anywhere from 1 to a few hours for the PCR, depending on the target. So you could run through that entire workflow within the scope of a single afternoon. And then the analysis, looking at the traces themselves, can be done, typically in about a minute per trace or so, depending on exactly what you're looking for. And that's kind of the value of having a more assay-specific software solution where you can automate that type of approach to be able to generate a lot of useful data relatively quickly. So even if you have a requirement to run hundreds of samples at a time, then the technology certainly accommodates that and can be formatted into those higher densities, and with the benefit of assay-specific software downstream that offers instantaneous automated genotyping, then you can generate a tremendous amount of useful information quickly to turn through those number of samples at a time. And the nice thing about the family of genetic analyzers and now the SeqStudio is that for labs that do have a need to look at fewer samples at a time, this being a 4-capillary system, then it accommodates that lower density very, very well, but then also gives you that flexibility to look at other types of run conditions, even comparing types of analyses on the same plate. So you can look at multiple small batches as well as one set of samples with the same set of conditions.
Thank you, Dr. Latham. And what was the sample input for the assays that you described?
So here again, having a lot of sensitivity to be able to detect low-level mosaic equivalence means that you can pick up the primary alleles from relatively small inputs. We use typically 40 nanograms of genomic DNA as the input into these assays. We can certainly use less, and in fact, part of our evaluation was to do a titration series. We were able to go down to much lower quantities in the range of even 1 nanogram or so, but our standard conditions are in the 40 nanogram range, and that's also to accommodate the ability to look at some of the lower level signals that may be present, so you can get a fully informed view of the information from the sample to support the clinical research questions you may be interested in.
Thank you so much. And thank you again for our audience questions. Our next one is, what run parameters did you evaluate during optimization of your assays on the SeqStudio?
So again, I think this is an area where the platform really shines because of the versatility it offers and how you can nimbly manage different sample conditions on the same plate. The standard way that one would approach the type of optimization on capillary electrophoresis would be to vary the injection voltage, the injection time, the run voltage, the run time. And so we worked with all of those variables to try to understand what were the right set of conditions for the given assay. And it absolutely depends on the assay. Some of the ones that I showed data from today were run for about 55 minutes as a run time. Some of these were as short as 20 minutes. Voltage conditions, well, I gave you the example of how changing the run voltage can favor either sensitivity or resolution, and depending on what specifications were, we can turn that knob. So we actually evaluated a broad sweep of run voltages from 4 kV all the way up to 13 kV in one case. And then we played around a little bit with the injection voltage and time as well. So I think within those set of variables, you have a lot of design space to be able to come up with preferred parameters for your given assay. And just working within those, we found that that gave us a lot of latitude for how we can make sure we're getting the best possible results that would allow us to accommodate the different needs of each of these assays.
Thank you so much, Dr. Latham. We have time for one more question from our audience members, and I just want to remind everyone that any further questions that are asked will be directed back to you via email by Thermo Fisher Scientific, our sponsor for today. Here's our last question, Dr. Latham. What was the workflow for the PCR-CE assays and time to result?
Yeah, so I alluded to this a little bit in the previous answer, but the nice thing about this approach is that unlike NGS, this is a very straightforward methodology. Take your purified genomic DNA sample. We've had experience with lots of different sample types just in general using this assay approach, from buccal cells to whole blood to urine to other sample types. And we can take that DNA, you put it into the PCR. PCR can be anywhere from an hour to a few hours, depending on what the target is and what the goals are. You take that product, you can dilute it directly into high dye. You don't need to clean it up. And then add in your ladder and inject, and then run it on the instrument. And again, in one of our cases with a Tom 40 assay, we ran as short as 20 minutes. So this means that our hands-on time is minimal, less than an hour for all of these assays. Even looking at larger batch sizes, it's very well accommodated within that workflow and requires very few steps. And as a result, we've had a lot of experience with folks running through these assays to get up and going quickly with minimal training and be able to run through either a small batch size or a large set of samples, even within the span of an afternoon. So I think that that's the real benefit of operationalizing the approach when you know what the target is.
Thank you so much, Dr. Latham, for your presentation and for your important research. I also want to thank audience members and LabRoots and our sponsor, Thermo Fisher Scientific, for making today's educational webcast possible. For more information, you can visit our sponsor at thermofisher.com. Before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through April 2018. You will receive an email from LabRoots letting you know that this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thank you so much for joining us. We hope to see you again soon. Goodbye.